Your lecturer is Dr. Richard Restak. Dr. Restak is Clinical Professor of Neurology at the George Washington University School of Medicine and Health Sciences. He also maintains a private practice in Washington, D.C. A prolific writer, Dr. Restak has written 20 books on the human brain, 4 of which were main selections of the Book of the Month Club. His awards include Georgetown University Medical School's Linacre Medal.
Listening to this lecture will change your brain. While you're hearing my words, your brain is shaping thoughts and images. You're remembering and associating what I'm saying with your experiences. You're forming new networks of ideas, which are encoded in your brain circuitry. Now, traditionally, we didn't think that way. We thought the brain was fully formed by adulthood, like all the other organs in our body. In fact, it is life experiences that shape and mold the brain throughout and influence neurotransmitters, which are the brain's messengers. Hello, I'm Richard Restak, Clinical Professor of Neurology at George Washington University School of Medicine and Health Sciences. And I've been studying and writing about the brain most of my adult life. My interest started in medical school, blossomed in recent years, as neurobiologists discovered that our brains continue to change throughout our lives, thanks to what's called plasticity. Now, in this course, we'll learn how you can take advantage of this exciting new research to optimize brain fitness. We'll explore various things, such as how you can increase your brain's power by your own effort. Apply what you learn about the brain to meet everyday challenges and enhance your quality of life. How to apply principles of brain operation to solve problems and learn new things. Now, in addition to mental exercises, we'll examine contribution of physical exercise to good brain health. We'll look at the importance of diet, the role of sleep and naps. We'll even have some fun with puzzles and games. Now, here's a key point that we should set up from the very beginning. Physical and mental exercises differ. There's an important difference between strengthening the brain and improving general body conditioning. For instance, if you practice any physical exercise, swimming, running, boxing, whatever you like, it'll lead to general health benefits, such as decreased weight, blood pressure will become lower, cholesterol should drop, your stamina should increase, and so forth. Mental exercises, in contrast, tend to be more specific. For instance, a memory exercise won't help you very much in scoring higher on a test of logic. The bottom line is that you'll achieve the greatest benefit by working on separate functions such as visual observation, or fine motor skills with your hands, tactile perception, logic, numbers, imagination, and visual spatial thinking. Now let's look briefly at 3 of the important functions that we'll explore in this course. The first is attention, and by attention I mean focus, keeping your mind on one thing at a time. Second is memory, general memory. And third is a special kind of memory called working memory. First, attention. Attention is the gateway to top performance in math, reading, auditory, and visual memory. It provides us with the basis for learning what to do and focus on and what to ignore. It's the coordinator of brain networks involving things like sensation, movement, emotions, and thinking. The second area is memory, which of course is an extension of attention. We can't use information that we can't remember. When we exercise our memory, we activate and maintain widely scattered circuits throughout the brain. In a future lecture, we'll discuss practical, easy-to-learn methods for developing a superpower memory. The third area is what's called working memory. And this is the most important mental operation carried out by the human brain. Let me repeat that. It's the most important mental operation carried out by the adult human brain. Now, we use our working memory when we simultaneously keep various multiple things in mind and sort of mentally manipulate them. Let's cover a few basic principles of brain operation. Let's first start with the frontal lobes, the CEO of the brain and the seat of what's called controlled processing. The frontal lobes can be linked with everything that distinguishes us from other animals. Included here are things like foreseeing consequences of our actions, sequencing things, executive control, and drive. It's the frontal lobes that are involved whenever we consciously think something out, like preparing our income tax, getting all our expenses and our income and everything and balancing it out and thinking about what we're going to owe when we pay. So this is what we mean by controlled processing. This usually means when we talk about controlled processing, we are talking about thinking. We use words like reasoning. Of course, that's much more respectable and a respectable way of thinking because we've been led to believe that we are primarily reasonable creatures. But of course, many of our actions aren't based on controlled processing, like reasoning, but occur on the basis of automatic processing. Things just sort of happen, like my walking across this room here, or eating a meal, or driving to the drugstore. With automatic processing, choices are made with really no sense of effort. Typically, they can't easily be explained to others, and we're not even aware of how we decided. Now, since consciousness isn't involved, our reasoning doesn't make it onto our mental radar screen. Let's talk a bit about the role of automatic processing in normal life. Its importance is that it frees up areas of the brain for other uses. Alfred North Whitehead, the philosopher, saw this, and he stated that, quote, civilization advances by extending the number of operations that we can perform without thinking about them. Now, we shift to controlled processing when we encounter something unexpected and have to think about it. Or we have to explain something to someone else. Teaching someone to drive is an example. It demands controlled processing. Now, balance, of course, is required. Too much automatic processing and we become impulsive and just simply act without reasoning and thinking. But too much controlled processing leads to a paralysis of indecision. We keep running over in our minds various scenarios that we can or can't do, and we really can't make up our mind about what we're going to do. A balance is also needed between cognition, between cognitive and emotional brain processes. You'll hear a lot in this lecture about the word cognition, which is a fancy word, really. It's shorthand for all of the mental activities that we do, such as thinking, remembering, calculating, daydreaming, all of these responses that occur to our inner and outer worlds. As we'll see in future lectures, cognition and emotion often overlap. They're not so separate. Many thoughts and experiences are accompanied by emotions. You pick up the telephone and it's the voice of an irritating salesperson. Compare that to picking up the phone and it's the voice of a good friend. You're not just responding intellectually, you're responding emotionally to these two different experiences. So the reasoning and emotional centers are linked in the brain. Let's talk just a second about what brain areas are key for cognition and emotions and the distinction between them. Automatic processing is centered toward the back of the brain, the occipital, parietal, and temporal lobes. Controlled processing, however, mainly involves the anterior parts of the brain, the prefrontal and frontal areas. Now, cognition, since it involves the whole brain and special senses, we have specific processes such as reading, writing, listening, and talking, which we can't localize to one particular part of the brain. Emotions are a little bit more localizable. They involve deeper brain structures forming what's called the limbic circuit and the right hemisphere. So how does the brain manage to integrate our inner and outer experiences? As you may already know, brain function is localized. We have the right hemisphere and the left hemisphere. And each of these have different specializations. And each lobe in the hemispheres is concerned with specific processes. But despite the multiplicity of brain areas and the functions, we experience the world as a unity, as you are now as you're listening to my lecture. It's all coming together in one unit. In fact, we experience what the filmmakers would refer to as scenarios. Sights and sounds and all other sensory inputs are synthesized into a unified experience, such as when you're driving to work. You're not hearing one thing, seeing one thing. It's all coming together into one total experience. Our understanding of how the brain binds our separate sensory inputs evolved actually from the early history of TV. Sound engineers worried that audio and visual signals wouldn't synchronize. Then came the accidental discovery of a 100-millisecond window within the brain that automatically synchronizes the signals. But beyond that window, that narrow 100-millisecond window, you have a badly dubbed movie. The brain automatically synthesizes sound and visual scenes into the unity of a movie that you're watching. Here's a key insight about how the brain works. It's a very insightful view that we're going to have about information, because information is the unit of exchange of the brain. Let me repeat that. Information is the unit of exchange. Messages are the token of informational exchange. Message transmission is the basic brain operation. Now here's another key insight. Large brain reflects small brain. In other words, message transmission occurs at levels ranging from the easily observable, me talking to you, to the microscopic. Now let's talk a little bit about brain organization. Is the brain one vast interconnected network of fibers, or are the nerve cells physically separated from each other? There's a great deal of debate about this in the early part of the 20th century. Ramon y Cajal, who was a neuroscientist and also a great artist, felt that the brain cells were not in physical connection with each other, whereas Camillo Golgi, who was an equally well-known neuroscientist, thought that it was like one syncytial net, sort of like a spider web. Well, no one was really sure who was correct. In fact, the Nobel Prize Committee of 1905 couldn't make up their mind who was correct and gave the Nobel Prize to both of them. Now, in the interval, special stains, special ways of staining neurons, reveal that neurons are separated from each other by a clasp, or what's called a synapse. The axon of one cell is connected to another, but it's not a physical connection. In fact, brain cells are connected a little bit like people on cell phones are connected. They're in touch with each other, but they're not physically connected. Also, there can be strong or weak connections caused by distance, interference, competing signals, and so forth. In addition, there's what's called entanglement. The neuron's branches are tightly entangled with each other. Think of the brain as a gigantic bowl of spaghetti, with each noodle highly branched and entangled, but not quite physically connected with the others. Message transmission between neurons takes place in two stages. The first is electrical. An electrical discharge is generated and travels along the length of the axon to the terminal at the synaptic space. The electrical signal then triggers the chemical stage, which is the release into the synapse of the neurotransmitter, which is a messenger molecule. The neurotransmitter enters the synapse, travels across like a ferry boat to receptors located on the membrane of the dendrite. Now, what are receptors? Think of receptors as dynamic proteins within the cell membrane of the neuron. Now, we say dynamic because they increase or decrease in number over very, very quick and short periods of time. For instance, an increase in number occurs in addiction, leading to a withdrawal reaction when the drug is unavailable. At such times, the receptors literally cry out for the chemical. The withdrawal reaction ends when the receptors decrease in number. Well, let's get back to the ferryboat trip. The ferryboat trip from pre to postsynaptic neuron takes 1/1000th of a second. Each neurotransmitter attaches to a special receptor, just like a lock would attach to a key, highly specific. One dendritic cell, then another is activated or inhibited from firing. At this point, communication reverts again to an electrical signal, which travels along the axon until reaching the next terminal where communication becomes chemical once again. There's a life cycle for neurotransmitters. After attaching to the dendritic membrane, the neurotransmitters are either reabsorbed by the presynaptic neuron, prepared for re-release, or they're broken down into smaller chemicals. Think of the brain as the summation of billions of interacting neurons influencing each other via the interplay of hundreds of neurotransmitters and their receptors. Now let's shift our focus just a bit from the molecular world of neurotransmitters and receptors to the scale of the whole brain. The brain is an organ that during my neurology training I've even held in my hands. It's possible to discern a history of our understanding of the brain, and physicians learned about the brain through the abnormal brain. That's because early research tools were dangerous to use in normal people. So neuroscientists learned about the brain from people with brain damage. Of course, learning about deficits does help to understand the normal function, but only up to a point. Now, thanks to newer, safer imaging methods, neuroscientists can carry out wide-ranging studies of the brain's processing in everyday life. We have to think of it in two ways. It's called structural imaging and process imaging. We talk about structural imaging, think carpenter. It shows the location where things are happening. Now, CAT scans and MRI, or magnetic resonance imaging as it's called, is an example of structural imaging. Process imaging, think electrician, activities. Process imaging shows electrochemical activity. That's things like functional MRI and PET. We have methods that illustrate shifts in neurotransmitters, ions, and magnetic fields. Now, imaging is good, it can do a lot, but it can't do everything. Let's talk about what it can do. It can measure blood flow, which increases as neurons become active, and we get what we call activation maps. If you did one right now, it would show parts of my brain that have the activation of thinking, my lips, my tongue, my hands, all that's activated. So you'd see an activation pattern of someone speaking. If we did an activation pattern of your brain, it would be someone listening to something and thinking about what they're hearing. Imaging can do other things, such as to serve as a marker for disease states. We mentioned addiction a few moments ago. It can also provide developmental profiles, such as adolescence. You can tell probably how old someone is within certain ranges by looking at the imaging. Now, imaging can't do certain things. It can't predict individual behavior or proclivities. You can't say whether someone's going to turn out this way or that way on the basis of a brain image. And it can't always optimize current treatments for brain disorders. Because remember, the individual regions work independently and they come together. In addition, the more abstract the concept, the harder it is to localize it on a brain image. Take courage, for instance. There's no area of the brain that's going to light up for courage or consciousness. This was what we think of as neuromythology. It's a little bit like in the 19th and early 20th centuries, they talked about phrenology, that one could tell what someone was thinking by looking at a part of the brain, how big it was, how big that part of the skull was. We know now that something like that just simply doesn't hold up. Here are some basic brain facts, some numbers to take in that are just truly extraordinary. The brain encompasses 100 billion neurons, with each neuron connected to perhaps 10,000 others, for a total of 1 million billion connections. Wow. It's hard to get a mental picture of a number like 1 million billion, so let's sneak up on it in stages. 1 million's easy. 1,000 times 1,000, about the size of a small city. A billion is 1,000 times 1 million, which is the size of 1,000 small cities or 100 very large cities. Now, to put 1 million billion in perspective, there are about 6 billion people currently living on our planet, which is a small number compared to those 100 billion nerve cells, and a truly paltry number compared to those million billion connections. Also consider the difficulty in tracing the nerve cell connections. The lowly worm C. elegans has a brain containing 300 neurons, so we're really way down there compared to what we talked about a second ago. And this has 7,000 synapses, so 300 neurons, 7,000 synapses. Sounds like an easy thing. Actually, manually tracing the connections without the help of a computer took 10 years to complete. In comparison, let's look at another simulation of nerve cell birth and growth, only this time we're going to talk about the human brain. Now, this video that you're looking at focuses on the dynamism of what's happening in a small section of the brain. It shows an interlacing of the neuronal fibers. It shows a vast interconnectivity at all levels. The bottom line here is what you do affects dynamic processes like this in your own brain. This is also the underlying theme of this course. This video gives you an example of why even computers will have difficulty tracing the neuron connections in the human brain. And sorry to say, there isn't a quick solution to the numbers problem. Sebastian Sung, who had something to do with that film that we are watching now, is professor of computational neuroscience at MIT, and he has this to say about human brain tracing. You'd need 100 million terabytes to store the images from the human brain. Even with 10,000 microscopes working in parallel, it might take 30 years to collect all the images. Analyzing the images could require a parallel supercomputer with millions of processors. So here's the dilemma. Computer counting results in many mistakes. And computers, they're not always reliable, and they can't accurately see the shape of the neurons because the neurons do vary considerably in shape. Manual tracing is incredibly time-consuming, even for a cubic millimeter of brain tissue, as we saw in this video. Here's the take-home message. Don't expect to see a wiring diagram of the human brain in your lifetime. Let's turn to brain development. The neurons are produced first, followed by synapses, which makes sense because you can't have a synapse connecting one neuron to another until you have some neurons. Synaptic connections form rapidly. In fact, in the womb, they're coming at 1 million per second. In the first month of life, synapses increase from 50 trillion to 1 quadrillion. Back again to these incredible numbers. Unused synapses are eliminated by pruning, which results in more efficient circuits. And pruning is just like you'd think about in a garden. You go out there and you get rid of things that aren't working, nothing's happening, and they're gone. Now, there are patterns of brain growth. There's a linear and progressive increase in brain weight, volume, and head size. Increasing numbers of synapses also come until a plateau is reached, followed by a decrease in nerve cells determined by usage and circuitry formation. The total brain volume peaks at age 11 in girls and 15 in boys. The male brain on the average, now we're talking on the average, is 10% larger. Brain reaches maximum weight by early adulthood and decreases 10% over the lifespan. So think of a 3-pound organ with 100 million nerve cells and 1 million billion synapses. It undergoes functional changes over the lifetime. Between 3 to 6 months, the brain achieves its maximum brain cell number. From 7 months to 2 years, the nerve cell numbers decrease, while connections or synapses increase. 40% of synapses generated in infancy are lost by adulthood. This is the use it or lose it theme, which goes throughout the whole study of the brain. Those parts of the brain that are not used are in danger of being eliminated. The brains of animals with enriched environments contain 25% more synapses, and we're going to go into that in the next lecture. Details about this in lecture 2 on plasticity. There's also patterns of brain growth with distinct peaks occurring at certain times, such as age 3 when you have individuation. Age 7, when the beginning of concrete reasoning starts. Ages 11 and 12, when you have the influence of hormones and the onset of puberty. At age 15, when abstract reasoning and judgment starts, when a person is thought to have both legal responsibility for their actions. Beyond 15, brain development depends very much on experience. With maturation, gray matter or brain cells decreases while the white matter or the connections increases. Myelination, which is a term for insulation of white matter, increases and progresses from the inferior part of the brain to the superior, from the posterior to the anterior. The white matter increase is associated with greater efficiency and speed of conduction. Particularly important with the frontal cortex and the cerebellum, which mature late, and there are intimate connections between these two. Now let's talk now about the cerebral cortex, which is the outer part of the brain. If you take a deck of cards, you take out 6 cards, 1, 2, 3, 4, 5, 6. They're essentially 2 millimeters in thickness, and they have 6 layers. Each one's different. Each one's interconnected. And that's what we're seeing with the brain. It has that same arrangement. To appreciate this, you just think about that, bring the business card. You can use business cards as well, and you think about it, or, as I did, a deck of cards. Now, stretched out, the human cortical sheet, which is what we're talking about, is about the size of a large dinner napkin. The monkey neocortical area is about the size of a business envelope. Now, beneath the cerebral cortex is the white matter, which is tightly bundled communication lines linking points of the brain. Think of that tightly wound fibers that you find in a baseball that are hidden beneath the leather covering. And in this case, the leather covering is the cortical mantle. Within this bundle of white fibers are the glial cells. The classical view was that the glial cells constituted a scaffolding for neurons. Glia means glue in Greek. Glia numbers outnumber neurons by as much as 6 to 1 in certain parts of the brain. So obviously the brain is more complicated than we imagined even a few years ago. Neurons at 100 billion are outnumbered by glial cells, and we're not certain at this point what the function of these glial cells is. They process information, we know that, but they do it differently than in neurons. They're slower, and they communicate broadly rather than linearly. So we have a new concept of glial cells, which are now known to be involved in message transmission. They don't generate electrical impulses, but they do respond to them. They use calcium ions instead of electrical signals. They possess sensors that detect signaling by neurons, and they can alter it. Now let's say a few words about the basal ganglia, which are islands of gray matter suspended in the white matter. So they're down underneath, they're islands, they're connected to each other, and they're connected to the cerebral cortex. The frontal cortex particularly is especially interconnected with the basal ganglia. They're concerned with organizing automatic rather than intentionally directed behavior. As I walk across this room, for instance, I have something in mind to get to something, pick up something. That's the frontal cortex. When I went to get those cards, for instance, the frontal cortex set that up. But each movement that I made was not being controlled by or thought out. So these circuits are extremely complex and convoluted with both positive and negative effects. So the brain is a work in progress, which is a new insight. New neurons are created over the lifespan. We have granule cells of the hippocampus and the dentate gyrus continue to be produced, and that's called neurogenesis, which can be increased by enriched environments or inhibited by stress or depression or sleep deprivation, anxiety, things like this. We also have an experience-dependent brain. Infants born with the capacity to perceive and react to the phonemes of all the languages of the world. So whatever language they hear, they can react to it. By 6 months, this capacity is lost. For instance, Japanese children, if they don't hear English, they lose the ability to make the distinction between R and L sounds. Exposure to language increases proficiency. 20-month-olds with talkative moms know 130 more words than a child with a laconic mom. At age 2, the number's risen to 300 words. And only live interaction with a person works, not TV. The child's third grade achievement is related to the amount of early language exposure. So you can see the importance of this. We have critical and sensitive periods. Kittens deprived of vision in the first 3 months of life, for instance, grow up with permanent visual impairments. And songbirds that do not hear their native sounds can't produce those sounds. Monkeys raised in isolation grow up with emotional problems. Among people with perfect pitch, this occurs only in musicians who started training before age 7. And in order to speak a language, a foreign language, without an accent, a child must speak the language's phonemes by puberty. But despite these sensitive periods and critical windows, the brain is still able to form new connections and form new associations, not only in adulthood, but far into old age. So what have we learned so far? Brain circuits and nerve cells can increase in number. Each brain is a result of individual experiences and choices. And the richer, the more varied the experiences, the more enhanced the brain function. In other words, what you do shapes your brain's structure and function. The electrical chemical activity in the circuits determines their fate. We say cells that fire together wire together. Information is widely disseminated among neurons, leading to higher order processing such as memory, imagination, and creativity. The important point is the brain remains modifiable throughout our lives. In the next lecture, we'll learn more about our ability to change our brains through experience as we examine brain plasticity in more detail. Thank you.